The overall goal of this procedure is to design and fabricate microfluidic devices for observing how cells respond to environmental stimuli.
These methods can help answer key questions in the cell biology and biomedical engineering, such as how cells respond under different environmental stimuli, including chemicals, shear stresses, and electric field. The main advantage of this technique is that the chips are easily to be fabricated and assembled. And these microfluidic devices mimic the in vivo condition with high throughput capabilities. Demonstrating the procedure will be Tzu-Yuan Chou, an undergraduate student from my lab, and Hsien-San, a postdoc from Dr. Tzu-Yuan Cheng's lab.
To begin, turn on the carbon dioxide laser scriber and check its connection to the computer. Then, load a pre-drawn Christmas tree pattern, like one of the patterns shown here, into the scriber. Next, place a PMMA sheet or a piece of double-sided tape on top of the stage of the scriber. Adjust the focus of the carbon dioxide laser using the calibration bar and the visible helium neon laser. Then, scribe the pattern onto the substrate. When finished, pick up the patterned substrate, remove the unwanted pieces, and use a stream of nitrogen to clean the surface of any remaining debris. Repeat this process for each component of the chip.